Hello, once again, viewers. Welcome to the Eastern Cape Department of Education broadcast studio. I am Mrs. Nkosha, a life sciences educator from Richard Vacha Technical High School in the Buffalo City Municipality District. Today, we are going to be looking at genetics. Now, our session is a continuation from the last one that we did on genetics, which was looking at the basic concepts in inheritance. This particular one, we will be looking at monohybrid crosses for your full understanding, and we will be covering 3 types of dominance or 3 types of inheritance. Now, from the example you have been given now, that use a genetic cross to determine the possible genotypes and phenotypes of the children of a heterozygous brown-eyed male. Now, when you look at a person, you can't see whether they are heterozygous or homozygous, so we do not include those when we are writing a phenotype, but it helps us to write the genotype. So what is the phenotype of the male? Brown eyes. So we have got a male that has got brown eyes. Now, this male with brown eyes is crossed with a female that has got blue eyes. So now there is the phenotype of our parents. And then from there, you need now to write the genotype. Now, it is where now the heterozygous and the homozygous come in to guide how you are going to write the genotype. Now, use a genetic cross to determine the possible genotypes and phenotypes of the children of a heterozygous brown-eyed. So it means that we know that there is this dominant allele for brown eyes. But then now, because it's heterozygous now, so it means you have got 2 different alleles, but this one is masked by the dominant one. Hence, it is not showing in the phenotype. Then blue eyes, now remember this is recessive, it only shows in its homozygous state. So now remember, after genotype, now we then represent meiosis, because now these gametes, they need to separate. And then now you have got gametes. The alleles need to separate when they are going into the gametes. So now for our gametes, now we have got B, we also have B, so these are the gametes of the male. These are crossed with the gametes of the female. So now let us do crosses to represent fertilization. So it means it is possible that for this allele, it can combine with that allele from the egg of the mother, and it will give us a heterozygous genotype. And then it is also possible for this one to cross with this one from the female. It also gives us a heterozygous genotype. And then now the second allele now from the male, it can cross with the first allele from the female, and it will give us a homozygous recessive genotype. And now the second allele from the male can cross with the second allele from the female, and it will give us also a homozygous recessive genotype. So what were we doing now here? This is fertilization. Then from here now, what is it that we are getting here? We are getting the first filial generation. So this is F1, and this is the genotype. Okay, so now when we look at this genotype, it means that now we have got from this genotype, we have got 2 heterozygous, and we have got 2 homozygous recessive. And then from this genotype, we can now write out the phenotype. So now, because of the presence of this one, then it means that these 2 individuals will have brown eyes. And now these ones, they have got recessive alleles, so it means that they have got blue eyes. Now, let's see now if we have answered the question. The question was requiring us now to use a genetic cross to determine the possible genotypes and phenotypes of the children, which we have done because we have now showed that in the F1 generation. Okay, now let us move on Remember, we had said that we are going to be looking at 3 types of inheritance. So the first type of inheritance we are going to be looking at is complete dominance. Now, what is complete dominance? Now, in complete dominance, as we have already said when we're looking at the law of dominance, we have got a dominant allele and a recessive allele, and in the phenotype now of the offspring, the recessive allele will not show, the dominant allele will show. That is complete dominance. Let us look at the examples that I have here in front of me. Now, this particular one, you have got here a plant that has got purple flowers crossed with a plant that has got white flowers. But then now all the offspring have got the purple flowers. So what does that mean? It means that the purple flowers are dominant, the white flowers are recessive, and now the recessive one is masked in the phenotype of the offspring. Now, let us look at the second example that we have here. We have got a second example here where we have got an individual that has got brown eyes crossed with an individual that has got blue eyes. But then now all the F1 offspring have got brown eyes. So it means that all of these ones, they have to be heterozygous. So it means that now what it is showing us here, it is complete dominance, that when you have got a recessive allele and a dominant allele, the recessive allele is masked in the heterozygous condition. It is only the dominant allele that will show in the phenotype of the offspring. Now, let us take an example. Now, the example that we have here, which is our genetic problem 1, under complete dominance, seeds can be round or wrinkled. So it means that now we have got 2 phenotypes, round or wrinkled. Use a genetic cross to show the genotype and phenotype of the F1 generation when 2 heterozygous plants with round seeds. So now, if we've got heterozygous and we have got round seeds, we know that the type of allele that is shown in the heterozygous condition is the dominant one. So it means that round seeds now have to be dominant over, wrinkled. Now, the allele for round seeds is dominant over the allele for wrinkled seeds. Use R for round and small r for wrinkled. Now, let us do our cross. Remember, always when you do a cross, the first thing that you need to do, you have to write P1. Now, out of the P1, you have to show now the phenotype of the parents. So now let us look, what have we been given? The allele for round seeds is dominant. Use these letters. Okay, so now use a genetic cross to show the genotype and the phenotype of the F1 generation when 2 heterozygous plants with round seeds are crossed. So it means that now both the plants that we are crossing have got round seeds. Now remember, in the phenotype, when you look at a plant, you just see round seeds or wrinkled seeds. You don't see homozygous or heterozygous. So hence we don't write that in the phenotype. So it's 2 plants with round seeds. And now both of these plants, we have been told that they are heterozygous. So it means that now for our genotype, now remember we're given the letters R and R. So if it's heterozygous, we are going to have 2 different R's also for this one. Then after genotype, remember, it is very important to label. I will also show you how the mark allocation is done, the importance of these labels. So it's meiosis. And then we have got gametes. Okay, now instead of the crosses that we did in the first cross that I showed you, there is an alternative where we can use what we call a Punnett square or a Punnett diagram. Now, what we can do here is that we can write the alleles of the first plant here, and we can write the alleles of the second plant here. And then now you can see that now what we are going to be representing here in the middle now is the genotypes of the offspring. So this R and this R, they meet here. So there is the genotype of the first possible genotype that we can have. And then this R and this R, they meet here. So there is our second possible genotype. And then the R there that is at the top with the R that is at the bottom, they will meet here at this box. So we have got another heterozygous one here. And then now, for this particular box, you can see that it is this R and this R that are meeting here. Then now you have got your fourth genotype. Now from here, you can then, so what were we doing here in this Punnett diagram? We were doing fertilization. Then from here, we will have F1, where now we will represent the genotype. So what do we have as the genotype? We have got one RR, which is this one, and then we have got two of these. So we have got two heterozygous and we only have one recessive. So we have got one recessive. So now, the only thing that's left for us to write now is the phenotype of the F1 generation. So now let's represent the phenotype of the F1 generation. Phenotype. Okay, so for our phenotype, we can see that now this one has got dominant alleles together with this one. So it means that now we have got three now that will have round seeds, and only one that will have wrinkled seeds. Now remember, we said that now in the phenotype, hence we are able to combine these three because all of them will give us round seeds. Okay, now let us move on now to the second example of complete dominance. Now, in the second example, which is genetic problem 2, it is saying in humans, the ability to roll the tongue is due to a dominant allele. A man who is heterozygous for tongue rolling and a woman who cannot roll her tongue have children. Use symbols T and T for the alleles of tongue rolling characteristic and represent a genetic cross to determine the percentage chance of having children that cannot roll their tongue. Okay, so what is the first thing that we do when we represent a genetic cross? We need to write P1. Now, in P1 now, we write the phenotype. So now, what is the phenotype of the parents? A man is heterozygous for tongue rolling. So it means the men can roll their tongue. So we have got a man who is a tongue roller. This man is married to a woman who cannot roll her tongue. So the woman is a non-tongue roller because she cannot roll her tongue. And then now we need to write the genotype. Before you write the genotype, look for the keywords homozygous and heterozygous. So now the man is heterozygous. We have been told to use T, so it means that our genotype will have two different alleles. And then the woman is not a tongue roller, which is a recessive trait, so it means that we have got two recessive alleles here. And then what is the next thing? Meiosis. And then there are gametes. Okay, let's use a Punnett diagram again. Okay, now let us represent the alleles for the male. It's T and T. Remember, they have to separate, law of segregation. And then the alleles for the female, it's T and T. Then now what will be the genotype of the first possible offspring? It will be heterozygous. The second one, homozygous. The third one, heterozygous, and the fourth one, homozygous. Remember, when you reach this step, you can either do this Punnett diagram or slash, you can do the cross as we did before. So you can write the alleles of the man and then the alleles of the woman. And remember, the first one, which gives us the same as this one. Okay, and then the second one, remember now, both alleles of the male have to cross with both alleles of the female to give us the possible genotypes of the children. And then we cross those. Then now we can see that this one, we have only crossed it once, so we cross it with that one. So as you can see, now we are getting the same genotypes for the F1 generation. So what were we doing here? We're representing fertilization. And then now for our F1 genotype. Okay, so for our genotype, we can see that we've got 2 heterozygous and 2 homozygous. So what does this mean? So now, what is the phenotype? It means that now these 2 are tongue rollers because of the dominant alleles. And then these 2 are non-tongue rollers. So it means if we simplify it, it gives us a ratio of 1:1. Now, can we look if now we have answered the question fully? What was the question requiring of us? The question said that we should use symbols T and T for the alleles of the tongue rolling characteristic and represent a genetic cross to determine the percentage chance now of having children that cannot roll their tongue. So it means we have not fully answered the question because there will be now that one compulsory mark that is for percentage chance. So let us just represent it here on the side, the percentage chance. So now for having children that cannot roll their tongue. So it means here we have got 2 non-rollers. So out of the 4 children, 2 of them cannot roll their tongue. Remember, for a percentage, we always multiply by 100. So it means there is a 50% chance of having children that cannot roll their tongue. So what would be the mark allocation here? So it means that the marker will first tick there for the correct phenotype that is written, the correct genotype that is written, the correct gametes that have been represented, the correct genotype for the F1 generation, and the correct phenotype. And then that will be 5 marks. Remember now the standard mark for a genetic cross in life sciences, as you can also see here, it's 6 marks. But because now this question has specifically asked us to determine the percentage chance of having children that cannot roll their tongue, so it means that the 6th and the compulsory mark would be the percentage chance. Then you will see a marker, we'll put a mark there with a star to indicate that this is a compulsory mark that you had to do because the question specifically required you to calculate the percentage chance. Now, let's say maybe you did a mistake and maybe did not represent the phenotype correctly for the F1 generation and you did not get that mark. So now the marker would have to look for other possible marks. In life sciences, if you have written P1 and F1, that is a possible mark. And also if you have written meiosis and fertilization, that is another possible mark. So it means that you can still get full marks even if you do a simple mistake in one, because there are more than 6 possible marks, but we can't mark more than that 6. And if now the question specifically required you to calculate percentage, then it means if you did not calculate the percentage chance, then you cannot get the full 6 marks. Okay, now let us move on. Let us now go to the second type of dominance, which is incomplete dominance. Now, in incomplete dominance, neither one of the 2 alleles of a gene are dominant over the other one. Now, remember, in complete dominance, we had said that one is dominant, one is recessive. But now, in incomplete dominance, we are saying neither one is dominant. And now this results in an intermediate phenotype in the heterozygous condition. So now what does that mean? Let's look at the examples here. Here we have got chicken. Now the first one is black. We have this black one crossed with this white one. Now look at the phenotype of the offspring. It is neither black nor white, but it is a bluish gray type of an offspring. So it means that now when you are looking at examples with incomplete dominance, so instead of having two phenotypes, looking for the phenotypes of the parents, there will be now a third phenotype which will be different from both of the parents, but it will be an intermediate phenotype, meaning it will be something in between the phenotype of the parents. Now let's look at the second example. Now in our second example here, you have got here a white flowering plant that has been crossed with a red flowering plant. But now look at all the F1 offspring. They have got pink flowers, which we did not see in the parents. So now what does that mean? It means that now both of those phenotypes or both of those alleles are not dominant. Hence now you are ending up with an intermediate phenotype. Now let us look at the examples of how you can represent crosses with incomplete dominance. Okay, we have got an example here. Our example here is saying a homozygous snapdragon plant with red flowers was cross-pollinated with a homozygous snapdragon plant with white flowers. Now, I want you to notice something here. Now, we have got a capital letter R with a capital letter W. We no longer have a capital letter and a small letter y. It is because now this is incomplete dominance. So it means neither of the alleles are dominant. So we'll no longer be seeing in examples of incomplete dominance a small letter and a capital letter, because the other one, it represents recessive and the other one dominant. So now neither one are dominant. So we'll represent both of them in uppercase letters. Okay, now all of the plants that grew from the cross had pink flowers. So what I want you to notice again, in incomplete dominance, you will have the phenotype of the offspring, of the parents. In this case, you have got red and you have got white. But then now all of the offspring now are having pink flowers. So you will always have a third phenotype, which is something sort of like an intermediate between the phenotypes of the parents now, which will indicate incomplete dominance, because in some exam questions, they might not necessarily tell you what type of an inheritance or what type of dominance the cross is representing. They will ask that, or they will require you to know it and give the correct answer. So it means that in examples where we have got incomplete dominance, you need to look out for the third phenotype, which is an intermediate between the phenotype of the parents. So now let us represent now this cross. Remember, always when we are writing a cross, you need to write P1. Because remember, if you represent P1 and F1 correctly, you get a mark. Okay? So the first thing that you write, again, phenotype. So what phenotypes do we have here? We have red flowers that are crossed with white flowers. Okay, then genotype. Remember, before you write genotype, you need to look out for homozygous and heterozygous. So a homozygous snapdragon plant with red flowers, so it is homozygous, so it is going to have two RRs, is crossed with one with white flowers, and we're given the allele for white flowers is W. So note there we don't have any small case letters. Then after genotype, always we write meiosis, because now there is segregation of the alleles when they are going into the gametes. Then we have got gametes. Okay, let us do a Punnett diagram. Then we have the alleles of the first parent and the alleles of the second parent. And we can already see that now all of the F1 generation will have the RW genotype. Okay, so here this is representing fertilization. Okay, then now remember, now you have got the F1 genotype. So now in the F1 genotype, all are RW. Then phenotype, we have already been given the phenotype of the offspring. We have already been given the phenotype of the offspring from the cross, because it had said that all the plants that grew from the cross had pink flowers. So it means that now this RW is the combination of the red and the white, which gives us pink flowers. So let's see if we have answered the question. The question was saying, represent a genetic cross to show the possible genotypes and phenotypes of the F1 generation plants. We have done that. Here is the genotype. All of them are RW, and the phenotype, all of them have pink flowers. Remember, this is 6 marks. Let me use a different colored pen to represent the marks. We've got a mark for the F1 phenotype, a mark for the F1 genotype, a mark for the gametes, a mark for the F1 genotype, F1 phenotype. How many marks do we have now? We have got 5. Then we can get the 6th mark from P1 and F1, and possibly another possible mark would have been for the mark for the meiosis and fertilization, but we have already reached the maximum of 6 marks. Okay, now let us move on now to the second type of the second example in incomplete dominance. Okay, now in mice, the allele for black fur is B. So underline here, very important. And the allele for white fur is W. 2 gray mice were mated. Now there is a third phenotype that is coming in, which shows that we are dealing with incomplete dominance, because gray is an intermediate between black and white. Some of the offspring were gray, others were black, and some white. So how is this possible? Do a genetic cross to explain this result. Remember, every time genetic cross, you represent P1, you write phenotype. So what is the phenotype of the parents? It is saying 2 gray mice were mated. So it means that up we have got gray fur and gray fur. Okay, the keyword there is gray. The mice appear to be gray. And then the genotype. Now, remember, now we can't introduce another letter for gray, because now gray is a combination of black and white. Hence, we'll use the allele. So it means for gray fur, we will have BW and BW. Okay, then meiosis. Then we draw our Punnett diagram. We've got BW as the alleles of the first parent and BW as the alleles of the second parent. And then gametes, so these alleles represent the gametes. Okay, so the first one now we will have BB, we have BW, BW, WW. Okay, so now here in the middle, now we have got the genotypes of the F1 generation. So now F1 genotype. Okay, then we write phenotype. Okay, then under genotype there, we have 1 BB, we have got 2 BW, and we have got 1 WW. So now the BB, it means that now go back to the question. It is saying when 2 gray mice were mated, some of the offspring were gray. Here we are getting the gray from the BW. Some of them were black. We are getting the black now from the BB. And we are getting the white from the WW. So now we have answered the question of how it is possible that some offspring are gray, others black, and some white. Okay, now let us move on to another type of dominance, which is the third one, because we have covered the complete dominance and the incomplete dominance. Now we look at the third and final one, which is co-dominance. Now, what is co-dominance? Now, in co-dominance, both of the alleles of the gene are equally dominant. So remember, a dominant allele, it will always show in the phenotype. So it means if both of them are dominant, it means that now both of them will be expressed equally in the phenotype, because a dominant allele is always expressed in the phenotype of the offspring. Now, let us look at the examples of the pictures that we have here. So now, when we have a cow with a red coat that is crossed with a bull with a white coat. Now we are having offspring that are roan. So what is roan? It means this is an equal distribution of red and white, because remember now both of the alleles are dominant, so both of them will show in the offspring. Let's look at another example, the second picture right here. So now in this one we have got fish. The parents, one of the parents is red, the other one is blue, and now in the offspring we can see an equal distribution of red and blue. So very importantly, you can see the differentiation between now the three types of dominance, because now in complete dominance, one is dominant, one is recessive, so the dominance shows in the phenotype. In incomplete dominance, neither one is dominant, so neither of those phenotype they show. Hence you've got now a third phenotype, which is an intermediate between the two. And then in co-dominance, because now both are dominant, so both now will show in the phenotype of the offspring. So let us look at examples now of crosses for co-dominance. Now let us look at one that has already been done here. This one is similar. Now in front of us we have got a worked out example for co-dominance. Now we have a plant with white flowers that is cross-pollinated with a plant with red flowers, but then now all the plants that grew from the cross had an equal distribution of red and white. We can see this example is similar to the one that we were given in incomplete dominance, but we can tell now because the difference now is the result of the F1 offspring. In incomplete dominance we had pink offspring, here we have got an equal distribution of red and white. So now for the co-dominance, now this is a take-home activity that you can do. Please remember that for both co-dominance and incomplete dominance, we use capital letters. Now this is a take-home activity where you can do this genetic problem, where a farmer has an orchard of apple trees. Each apple produced expressed red and white color equally. To extend his orchard, the farmer collected seeds from the red yellow apples and grew them. When the new trees matured, he found that some of the trees produced red apples, others produced yellow apples, and the rest produced apples that were red and yellow. So you can use a genetic cross to explain this result in the F1 generation. And if you have any questions, you can leave them in the comments, or also you can leave your worked out examples in the comments. We have now come to the end of our lesson. You can leave your comments. Don't forget to leave the name of your school so that we can be able to give a shout out. Please share, recommend, and subscribe to our YouTube channel. And thank you for watching.